First we'll start with DNA repair. So first we have to talk about mitosis. That's when DNA replication occurs, and that's where a lot of the DNA damage or kind of the mismatch and mutations may occur. It can occur through outside forces, through damage, which we'll talk about more, like radiation or ingesting or inhaling external carcinogens or mutagens. But mainly, a lot of it comes naturally during DNA replication in mitosis. So during mitosis, there are multiple checkpoints in place that ensures not only the cell is growing properly and is ready for replication, but that during replication, there are no errors that occur which could lead to significant issues in the organism. The main one that many of you are probably aware of is cancer. Cancer is a genetic disease, and it happens through mutations in cells that get out of control, that aren't picked up by these checkpoints, that aren't picked up by a lot of these genes, and it kind of goes around them, and then it gets kind of out of hand. More mutations pile on and on and on, and that's why you get advanced cancers, and eventually people die of cancer. So there are three major checkpoints in the cell cycle. There's the G1 checkpoint before DNA synthesis begins, and this one is just to make sure that the cell is growing properly, right? This is at the end of G1 going into DNA replication. This just makes sure that the cell is growing properly, everything's good with the cell inside, and it's ready to divide. Then there's the G2 checkpoint before mitosis begins. So this is after the DNA has already replicated, and now the cell has two copies of all its DNA information, but the actual mitosis, the actual splitting, hasn't begun yet. And this is at the end of the G2 phase. So this one is to make sure that the DNA is replicated properly, there's no mistakes, that everything is set and nothing's missing, there's no extra chromosomes, everything is perfect, and it's ready to begin division into two cells instead of one. Okay. And then there's the M checkpoint in the middle of mitosis, and this one again is just to make sure that, again, everything's splitting properly, everything's attached to the spindle, and it's ready to begin pulling apart, right? Now let's talk actually about the DNA repair mechanism. So these are about 4 different types of DNA repair mechanisms that our body has. The first, and we'll be talking about each one individually. This is the one that's most common, is right here, DNA replication stress, so errors in DNA replication. And this leads to things like base mismatches, insertions or deletions, or something like that. And this is fixed via the mismatch repair pathway. So the mismatch repair pathway, this is the most common, as I said, most common DNA damage you'll see on the MCAT. And it's very simple. It's just, as you remember, there's meant to be an A and T pairing and a C and a G base pairing for DNA strands. However, there may be an error during replications that creates a different type of pairing. For example, AC or AG or TC or something like that. Or they may be, for example, you know that in DNA, you're never supposed to have U. Uracil, you're only supposed to have A and A, T, C, and G. So, for example, that could be something, or there may be an insertion or a deletion somewhere. Okay, and this just makes sure that this error gets stopped immediately and doesn't go any further. Okay, so you remember DNA transcription happens through an enzyme called the DNA polymerase. And DNA polymerase has the ability to detect these mismatches and repair them, sort of like a proofreading function. That's what it's called in a lot of textbooks in chemistry and biochemistry courses. It's a proofreading function. Some DNA polymerases have it, some don't. The one that humans have that we mainly use, and there are some that that bacteria, prokaryotes have, also have them. Some don't, polymerases. But those have different functions, and we won't get really too much into that. But the main DNA polymerase has this proofreading function. So what this means is as exonuclease activity, exo just means at the ends, and nuclease means it cuts nucleotides. So if you are going through a DNA strand, through a line, and then it notices there's an error somewhere at this end, and this base pair doesn't match up quite well, it'll just use the exonuclease activity, and it'll just go backwards, kind of like essentially like a backspace on your keyboard, and just go backspace, backspace, backspace a little bit and delete those last couple of nucleotides. that it created, delete the mismatch, and then it'll just kind of try retyping it out and re-putting it in, because it does. DNA polymerase obviously has the ability to put in nucleotides into the growing strand, so it just tries again essentially. And that's followed by ligation, because it does also have a ligase activity, which is essentially like a glue. When you put in a nucleotide, you need to actually attach the nucleotides to each other horizontally. So that's what ligase does, and that's called ligation. That may be a little bit confusing, but let them show you in a picture. So my S beta, these are just parts of the DNA polymerase that you don't really have to know. These are not, at least not for the MCAT, but for biochemistry courses you do, if you've taken those, but for the purpose of the MCAT, you don't. This is where the mismatch is, right? What it does is it creates a break, essentially right here, creates a break. Then it just kind of deletes using exonuclease activity, or just excise and just backspaces a bunch. And then it just followed by resynthesis, just remakes it. And then you have this little gap, and then ligation, mismatch repair. Okay, so the MUT-S and MUT-L locate, detects it. DNA polymerase comes in with the pCNA as the ring, the RFC binds to it as exonuclease activity, so it just cuts, right, like kind of like Pac-Man. You can see right here, just cuts it up, and then it religates it using this Pol delta pCNA, just remakes it. Then ligase comes in and ligates it. Okay, that's fairly simple, and that's about to the extent that you have to know. Obviously, it's way more complicated than real life. Like, you have all these like different polymerases, Pol delta. You have this pCNA, this ring. You have the RFC, which is very important. You have these different identification detection factors like MUT-S, MUT-L. Those are all very important, but you don't have to know that for the purpose of the MCAT. That's more of a biochemistry thing if you really wanted to go deeper. Okay. Next up is base excision repair. So sometimes things like radiation, free radicals, oxygen radicals, or other external factors can damage certain nucleotides on a DNA strand, turning them into an unusable base. Like, for example, you can turn guanine, the G, into 8-oxoguanine, which is not exactly the same as guanine, and it has different properties. And or you can turn it into an alkylated base instead of just a regular base. And these base pair with either the wrong nucleotide, which creates a mutation. So, for example, if normally you had, let's say, G, 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 and that obviously base pairs with C, 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 but you had now G, OG, which is oxyguanine, and G. What this does is it turns this into C. This base pairs with, I believe, T, and then C. Now you turn the CCC to CTC. And when this strand will be replicated, this will now be CTC, and now it'll be GAG. So, because remember, these are two strands, and then during the next round of DNA synthesis, these will split apart, and each of them will be used as a baseline and to create a new copy. And so what you started with was CCC GGG. What you end with is CTC GAG in some DNA strands. That's a point mutation, right? And we want to get rid of that because that could lead to something wrong. For example, if this happens in an important part of the coding sequence, for example, in the initiation sequence, the termination sequence, or anywhere else, something that's important functionally, that could change the complete function of the protein that's supposed to be made. It could prevent the protein from being expressed. It could create overexpression, something like that. Okay, so we want to avoid these point mutations. And this is caused by external damage, so radiation, free radicals, which you sometimes ingest or get exposed to, or other external factors. So how does this get fixed? It's by this mechanism called base excision repair, or BER. Once the damage is noticed by a protein called a glycosylase, an endonuclease will come in and it will create a tear in the damaged strand. An endonuclease is different from an exonuclease because an exonuclease, just like exo, it comes at an edge. So it can only start at one end of a DNA strand and go one way, essentially only this way. Whereas an endonuclease can come in the middle of a DNA strand. So it can come in like right here, then it can go come in the middle of a DNA strand, it can go this way, or it can go this way. Okay. And it has the mechanism on how exo and endonuclease work is different, but you don't really have to know that. So remember, exo only comes in at the end of a DNA strand and works its way down. Endo can come in the middle of a DNA strand and cut out just a small piece in the middle. Okay, creates a tear in the damaged strand. This will then attract a polymerase to come in and resynthesize a chunk of the strand, just like we talked about before, a DNA polymerase. Then the flap created will be excised by a flap endonuclease, then a ligase to patch everything up. Again, a lot of words. Let me just show you a picture to make it easier. You have a damaged base right here. Glycosylase comes and it recognizes that there's a problem here. Endonuclease comes in. It creates this little flap, right? You can see this flap, which it kind of removes this base. If it's a short patch, so say like a couple of nucleotides long, it will just get rid of the flap and it'll just put in something else, like using the DNA polymerase and then ligase. If it's a longer patch, like maybe 10 to something or nucleotides, it'll just come in, polymerase will come in and it'll go forward in this direction. You'll essentially have this where this flap is hanging, and this is synthesized. Then flap endonuclease will come in, get rid of the flap. So essentially all you're left is this. Ligase comes and patches up this little hole right here, ligates it. Okay. The overall process is very simple as far as the MCAT is concerned. You don't have to know a lot of the exact proteins. You probably don't even have to know that it's a glycosylase. The only thing you'll really have to know is that it uses endonucleases and then polymerases and ligase in the end. That's essentially all you really have to know. It's not important, but just in case you want to know it, yeah, it's helpful. And if you've taken a biochemistry class, you've probably already covered this and you know a lot in a lot more detail than it's needed. Next up is another process called nucleotide excision repair, or NER. There's some external factors like a UV radiation or polyaromatic carbons, which are found in things like coal, gasoline, tobacco. They're released upon burning these substances. That's why people work with a lot of coal, and especially near burning of a lot of coal, or people who are chronic smokers of tobacco, they'll end up getting cancer much more often, because when you burn these things like coal or tobacco, you release a lot of these polyaromatic carbons into your system, and these polyaromatic carbons can attach onto your DNA, and it can be mutagenic and in turn carcinogenic, because they can create damages in your DNA strand, which cause a lot of mutations or a lot of errors, and that's how you get the mutations that could possibly lead to more cancer. And these give larger lesions of damage in the DNA, more than just one or two bases, right? So they give big chunks of damage to one DNA strand. And this is important to note, and we'll cover it for the next type of repair. This base excision and nucleotide excision repair is when there's damage to one of the two strands. So right here, there's only damage to this one strand. Okay, that's really important. Okay, it requires a very similar but slightly more complex mechanism to fix, and it's called the nucleotide excision repair. Once the damaged site is identified by certain proteins, unwinding proteins are called in to unwind the double-stranded DNA and excise a large chunk of it, and then polymerase is recruited to rewrite the chunk. Again, very similar. Again, I'll show you a picture. So essentially you have damage. This over right here on the left is base excision repair, and again, short patch like one nucleotide, long patches up to like 13 nucleotides, whereas nucleotide excision repair can be like 20 to 30 nucleotides in length, right? But they're very similar processes. You have a small lesion, you have DNA glycosylase that comes in as the recognition, recognizes the damage, calls in something like an endonuclease to fix it. Polymerase comes in, fixes it. Whereas with nucleotide excision repair, so we're talking about, there's a big bulky lesion, a big lesion of damage. They call in these recognition sequences that come in, they bind, they actually unwind it like this, right? You can see here it just deleted a chunk, and for basic excision repair, for nucleotide excision repair, it unwinds the 2 strands first. Then it just takes a big chunk of 1 strand out, the damaged strand, and then the polymerase comes in and just fixes it. Okay, very similar. It's only really more complicated if you have to learn the exact steps and the exact mechanism, which you don't have to know. So you don't have to like recognize these proteins like the XPC or the TF2H or whatever. At least not the MCAT. Okay, those are the types of mechanisms we have for single-stranded damage, but some major damage can also be caused by radiation or other harmful chemicals called a double-stranded break. This means that both strands of DNA have a break in the same spot, where they have damage in the same spot. Since there is no longer a blueprint or undamaged second strand for polymerase to build off of to repair the damage, we need something else. Notice how both for basic excision repair, nucleotide excision repair, and mismatch repair, both of these use polymerase, DNA polymerase, to kind of just rewrite it and try again. And they do that because they can read this second undamaged strand and use it as a blueprint to retry and recreate the original strand. But if both of the strands are damaged, you can't use DNA polymerase anymore because you have nothing as a blueprint, right? DNA polymerase can't know to add an A or a G in that spot because it doesn't know that an A or G is supposed to go there because there's no C or T on the other strand that is telling you, okay? So we need something else, and this solution is called homologous recombination, and it uses the second set or the homologous pair double-stranded DNA as a baseline. Remember, you have two homologous pairs of DNA, one from your mother, one from your father. That's just how all organisms work, right? So you have this double-stranded DNA, and it had a break, as you can see right here. And you have this homologous recombination. You have these mediated proteins, but again, you don't have to memorize these at all. First thing you do is strand invasion. It tries to, for example, for this section, DNA polymerase has no idea what to do because it has no blueprint. But if you take this, you move it like this, and this is the area that's damaged, that has no second strand to build off of. If instead it unwinds the second homologous pair of double-stranded DNA, kind of moves it open, which is right here, strand invasion, right? This blue one gets a little bit cut up, like a little bit moved out of the way. This red one takes its place, and now the red one has something to build off of, right? This is supposed to be essentially the same genes as this red one, but just from a different parent, right? And it's just hoping that it's similar enough that it can copy it, right? And it tries to, that's exactly what it tries to do. It tries to copy off this blue strand as much as possible, just to repair itself, and then eventually it'll recombine with its other end. And meanwhile, while this strand, which we'll call strand 3, has been moved out of the way to make room for the red strand, it's actually being pulled up to the first strand, strand 1, and now strand 1 is also copying it to fix its own damage. What this creates is, and I'll show you here using the same colors. This was what it was originally. The damage looked something like this, right? What you end up getting is now something like this, right? So it has its own red DNA that it started with, but instead of having the other red DNA here and using it as a blueprint, it now has a copy of the blue DNA here and here where the damage originally was. Most of the time when you have a double strand break, this is kind of the best you can do because you don't know what information was originally there. That information is kind of lost. So the best you can do is try and take from a homologous pair and hope that it's close enough. This homologous recombination process, is also very important in reproduction. When you have kids and the baby is getting DNA from the mother and the father, the DNA from both parents actually undergoes this exact same process called homologous recombination on its own, because as you can see here, this strand is a little bit different than if I just had the original parent like this or the original parent like this. This creates genetic diversity by mixing and matching the parent's DNA, right? This is one of the reasons that we have so much genetic diversity, is because when we are born, we go through homologous recombination, which creates kind of a mixed genome of parents, right? It's a mixture of the genome between the parents, right? So you end up with some of this. Okay. So just remember, homologous recombination, very important for double-stranded breaks, and it's also used during fertilization for reproduction to create genetic diversity. The alternative to homologous repair is something called non-homologous entraining, and it doesn't utilize the homologous pair of double-stranded DNA. For example, if it's not available or whatever the reason is, if you don't have a second pair of double-stranded DNA, you go through something called non-homologous end joining, and it simply just takes the double-stranded break and glues the ends together. This is not ideal, as it means there will be a piece of lost information that was deleted from the DNA strand, and I'll show you an image. So pretend you have this double-stranded break. Instead of using double-stranded DNA or homologous recombination to do this, instead, you'll just take these chunks and kind of move them closer together and just stitch them together, and it'll be just a little bit shorter DNA, right? So, but this whole chunk, which used to be DNA, used to be information, is now permanently gone. This is kind of bad because that could lead to things like frame shifts, because if there's less than 3 or a number that's not a multiple of 3 that got deleted in this double-stranded break, it could lead to frame shifts, which change the whole sequence and the whole codon sequence, which could lead to a completely different protein than was intended. It could also lead to different kinds of point mutations or something like that. Overall, it's not ideal, but it's still better than nothing, because if you have this break, you just can't read the DNA anymore, and you can never make an RNA or protein out of it. So having this is like sometimes better, I guess, than having nothing, but this is really not ideal. Ideally, what you want is homologous recombination.